Hello, this is Craig Camp, and welcome to Troon Talk, where we dig deeply into Demeter biodynamic and regenerative organic certified wine growing and farming at Troon Vineyard in Oregon's Applegate Valley. This is meant to be a podcast for those that aspire to, as the Regenerative Organic Alliance says, farm like the world depends on it. These interviews will be focused on our work here at Troon Vineyard, but will also include the work of other farmers committed to regenerative agriculture. And now for today's interview.
Hi, I'm Nate Wall, the winemaker here at Troon Vineyard.
And I'm Micah Wagner. I'm the assistant winemaker here.
And today we're going to be talking about red winemaking, in particular some of the steps that are involved in red winemaking, along with the decision points at each of these steps. And in particular, we're talking about different products that may or may not be used at each of these stages in winemaking. You may have already listened to our previous podcast, which was similar information detailed for white winemaking, which is similar but different. And so today we wanted to get more detail onto the steps involved in red winemaking.
Yeah, so let's get into it, I think. To start, we will go over the different steps that we have broken this down into. And that way, hopefully, as we go, we know we can chat a lot, so you can kind of follow along. So step 1 will be harvesting, the picking of fruit. Step 2 is going to be fruit intake. Step 3 will be fruit processing. And then we move into step 4, which will be cold soak and fermentation. And then into 5, which is more of cap management and possible nutrients. Then step 6 is pressing and settling. 7 is malolactic fermentation. 8, we have racking into aging vessel.
It's possible those two steps may be reversed in certain circumstances.
Yeah, we'll go into a little more detail about a possible 9th step as well, which is racking off lees. And then we get into 10, which is aging. 11 is blending and stabilization. And then we have our fining and our filtration for step 12 and bottling finally for 13. So, Nate, I think you can start us off at step 1, which is harvest.
Yeah, so harvest is when you're going to be, as a winemaker, out in the vineyard trying to figure out that all-important picking decision. Like, when do you decide to pick those grapes? And you'll be sampling the grapes in the vineyard and probably bringing them back to your lab. You can sample individual berries from multiple clusters of grapes, like by multiple I mean hundreds. Or you can sample individual grape clusters. There's pros and cons for different sampling methodology, but in general, you're just trying to figure out like what is the chemistry of those grapes right now. What is in particular the Brix, which is the sugar levels, as along with the pH, the acidity, possibly the TA, the titratable acidity, and of course you're also checking for like flavor and in particular like phenolic development in red grapes, the tannins both in the skins but also in the seeds, like how ripe the seeds are inside the berries.
Yeah, so this kind of depends on what variety or varieties you're working with and what your targets are, but you're kind of planning based on your winemaking style. And so Nate touched on a few things, including Brix and pH, which are most common for people to be recording up to harvest. And if you are not adding anything, then you kind of have to make a few calls here. So here at Troon, for example, making more low intervention wines, we're pretty much picking on pH. And of course, Nate and I are also tasting this because, sometimes You take samples a few different times. They might look a certain way, but then you actually get the fruit in and it could be a little different. For other people that might be adding a few different things, you could be picking kind of a combination of what works best depending on what you're going to be adding. So you're not just making a call based on when the pH is ready, but it's more of a combination between pH and Brix, which again is sugar.
Yeah, and we'll get into the importance of why we use pH a little later on in some of these steps that we discuss. But in particular, another thing that may occur, and this is some of it's actually a little new, is field additions. So some people may choose to actually do additions to the freshly picked fruit, like in bin or in gondola, before it even gets to the winery. And in particular, I think an interesting thing that has recently started is inoculating with a non-Saccharomyces yeast in the vineyard, basically directly. This is particularly used, I think, in wineries that are purchasing fruit that comes from a significant distance away from the winery. I mean, at Troon, all of our grapes are coming from minutes away from the winery. They're all estate grown on site here, and so transportation isn't really an issue for us. But if the grapes do have to travel for long distances, some winemakers are choosing to add a non-Saccharomyces yeast in the vineyard to allow for a certain amount of, we'll talk about non-sac yeast in a bit, but to allow for a certain amount of protection to those grapes as they are transported, what could be hours, especially if you don't have temperature control during that transport. If it's like on a flatbed as opposed to like a reefer truck, fermentation could happen. Spontaneous fermentation could happen, and in a more conventional approach, they're trying to delay and or completely prevent spontaneous fermentation, which is, of course, the opposite of what we do here. We are all spontaneous fermentation, but they may add some non-Saccharomyces yeast. They might also add SO2 directly, sulfates directly in the vineyard. They could also add some enzymes, in particular like a lysozyme or something that could basically disrupt certain bacteria that could ultimately cause a problem in their fermentations later.
Yeah, and again, to be clear on that step, you would communicate with your viticulturalist or someone else at the pick, and you'd have some method to where you're picking and you want to kind of evenly distribute this in the bin so that it's actually touching all the fruit, and then you would transport like Nate said. So once everything is harvested and brought to the facility, you do your fruit intake. So that's typically weighing out your fruit, and just for reporting purposes, this is really important. But also, if you're not sure what tank a certain thing is going to go into, this tells you how large of a vessel you need. It also might tell you what your ads are going to look like if that is something that is in your program.
Yeah, so getting those weights, obviously, so you can estimate the fermentation, the fermenter space, like Micah said, is important. Often, and you have to report the tonnage of grapes harvested to the state that you're in. Some states require chemistry reporting as well, so it's not just the tonnage that you harvest, but the sugar levels or the acidity levels of those grapes sometimes need to be reported, and so that's the sort of thing you would be paying attention to. The main addition that some people might do at this point, and I think is very commonly done at this point, would be, especially if they hadn't done it in the field, they would do an SO2 sulfur addition to the grapes right now. And the main reason that a more conventional approach would do that is they are not planning on spontaneous fermentation. In fact, they want to prevent that spontaneous ferment because they're planning to inoculate with yeast, very certain yeast or yeast strains that they have very known characteristics, and so they want to prevent that and any other microbial growth. So a dose of SO2 directly at fruit intake is very common in the wine industry. Not anything we do at Troon. At Troon, we specifically don't add sulfur. And in fact, it's right now it's middle of May, and some of our 2024 wines have yet to receive any sulfur at all. So yeah, we delay the addition of sulfur as long as possible, and certainly not right at intake, because we want to encourage the microbiome to start getting active and doing its thing. But in a more conventional setting, they would want to suppress that.
Yeah, and so that kind of takes us into step 3, the fruit processing, because sulfur can be added to bins directly upon fruit intake, or also you can be doing it when you process and be measuring out depending on which vessels and how many grapes are going into each. But here we do quite a bit of whole cluster, and we also do some destemming. We have a setup that can slightly be altered depending on which we're doing. But for red intake, we are either sorting the whole cluster, just kind of, going through them, making sure there isn't any disease, mold, anything visual on the grapes, because again, we've mentioned we're not adding any sulfur. But typically people do have that option of either, they can directly put whole cluster into a vessel that it will be fermented in, or they can actually take a little extra time to look through. Same goes for destemming. So when we destem our red fruit, it goes onto a sorting table. And this is actually, I think, a great time during vintage at any winery, where it's like people will be around the sorting line, and a lot of times other people in the company can come out, join in, kind of talk to everyone about what's going on that harvest. I've had a lot of good conversations during processing at different places. So, it can be fun, like counting how many bugs come in on different crops of fruit, and just kind of getting involved, being a part of it. But whether we destem or are doing whole cluster, again, with our red fruit, we have that set up to look through all of it prior to it making its way into a bin, which then can either get put into concrete or amphora or one of our fermenting bins.
So the whole cluster that Micah just mentioned is a important consideration for us, going back to that picking decision with pH. When we use whole cluster here at Troon, and we use a lot of whole cluster, we like it. One is because we are able to do so. All of our grapes are manually harvested, which means a human being actually cuts each cluster of grapes off of the vine, which means that all of our grapes are harvested as whole clusters. Anything that's mechanically harvested, the mechanically harvested fruit is basically destemmed on the vine. So mechanically harvested fruit only harvests the berries, and so if people who are using mechanical harvesters, they do not have the option of doing whole cluster, because the rachies, the stems, are left attached to the vine. Only the grapes are removed. So that's actually a specific benefit of harvesting by hand. Not only is it, of course, a little more gentle than all these little fingers whacking the grapes to have them fall off the vine, but it also allows us to choose to use whole cluster or not. And so the use of whole cluster can do a lot to wines. I'm not going to go into that in this podcast. We've talked about it before, and we'll talk about it again. But one other thing it does that is relevant here is the more whole cluster that is included in a fermentation, the more that the pH is going to shift upwards, meaning the wine is going to become less acidic the more whole cluster is used inside that fermentation. And we know we like using whole cluster, and in particular, we know certain varieties like it more than others. And so going back to that picking decision, we need to ensure when we pick the fruit at Troon that we have enough acid basically in the picked fruit to allow for that shift that's going to happen later down the line. Other wineries, and actually this would happen right now, it's very common, most wineries would be adjusting their chemistry at this fruit processing stage. So they'll be either saignée the fruit, which would be draining off some of the juice to further concentrate the amount of juice basically the amount of juice that's in contact with the skins to have a more concentrated wine. If their sugars are too high, which is most places that are harvesting for bricks would probably have relatively high sugar levels because they're going for a certain kind of very ripe fruit flavor. That would also lead to a very alcoholic wine, so they might want to adjust that down, and the main way of adjusting it down is to add water to water back the fruit. And the same places that would need to do that would almost always have to add tartaric acid. I would say I can only imagine. I bet you tartaric acid is the single most common additive now used at fruit
probably.
Processing, right? Because
right after, we should say.
Right, yeah.
Once you get the right sample.
Yeah, right. Yeah, once you have the good numbers, because as the climate is, warmer climates basically lead to less acidity in picked grapes, and so adding acid to kind of counteract that loss of acidity is a very common occurrence. And what's probably a little less common now, but still definitely done, is this process, chaptalization, which is just the addition of sugar to the juice or the must right before fermentation, where you're not adding it for sweetness, you're adding it to increase basically the sugar levels of the wine, which will ultimately increase the alcohol level of the wine. This was much more typically done in very cool years where grapes struggled to ripen. That is becoming less and less of an issue in most wine regions of the world nowadays. But this would be an addition that would be done kind of at this fruit processing time, particularly after you get those readings. You want to talk about the readings?
Yeah, so a lot of the time, if you're working at a place that's going off a little more conventional winemaking protocols, you're going to be doing some of these ads, right? And so you don't want to immediately get in fruit, process everything, and take a sample out of the tank it went into, because say it's like a 2,000 gallon tank and you put a ton of fruit in there, and yes, it all got processed, but it's still pretty intact. So whatever sample you're going to get off of the sample valve, or whichever valve you're taking it off of, that's really not an accurate reading. You're going to want to wait a few days. Like, there are even places that you'll process everything into a tank and they'll immediately have you do a pump over on it, just in attempt to kind of start that process of the cold soak and getting a little more juice out of it. But you might also be wondering, it's like, well, why don't some places just pick off of pH, like we're mentioning, not to add other things? This is a stylistic choice. There are spots that say, okay, I don't like the taste of my fruit, I don't like the concentration at this certain pH. It's also based on, again, variety and where you're growing your grapes. So sometimes in one area you can get certain numbers that you can't get in another region. So if somebody really wants a certain concentration and a certain brix off of their wine, off of their juice initially, then they're going to have to decide like what sacrifice kind of to take. So it's like, do you harvest based off a certain pH but then add sugar, or do you harvest off a certain brix but then adjust the pH? So that's something a lot of winemakers work towards. Again, not everyone is so lucky to be able to just pick and not adjust. So that's maybe a topic to get more into in another podcast, but just in case anyone was wondering why you would do that.
Quick plug for the Applegate Valley. I mean, one of the reasons that we love this wine growing region so much is that, going back to the loss of acidity, acidity is lost, as I mentioned, kind of with higher temperatures. You lose more acidity. What we have here in the Applegate is this really good diurnal temperature fluctuation, meaning we have warm days but very cool evenings. And so the warm days, the plenty of sunshine, is really great for ripening the sugars as well as the flavors, the phenolics, the tannins in the grapes. But then those cool evenings and cool mornings are really good for preserving the natural acidity of the grapes. So it's much easier for us here to make that decision to pick on pH and to have the acid to do the things we want to do in the wine, like being able to use more whole cluster, and just at the end of the day having higher acids, which means lower pHs, which means a more stable wine. Particularly that a large part of why we can make the decisions we make is because of where we're growing these grapes. Like Micah said, you have to make different decisions sometimes for different regions, based on the outcomes you're looking for.
Yeah, so that kind of brings us into cold soak pre-fermentation, which...
So cold soak is, what is cold soak? Let's start with that. So cold soak is a period of time where the fermentation vessels are kept cold one way or the other to prevent the onset of biological activity, to prevent basically fermentation and or any other microbial activity from kicking off. And that can be done for a variety of reasons. It can be done there's certain things that basically can be extracted better in a more aqueous solution, like when there's more water available, like as juice. And there are other things that are extracted when alcohol is present, kind of at the end of fermentation, and then throughout the whole fermentation process. So a cold soak allows more time on skins for certain varieties that, for example, with Pinot Noir. Pinot Noir is a very thin-skinned variety. It ferments very quickly. And so it's very common that people will initiate a cold soak on Pinot Noir to be able to have this time on skins for various, certainly color being one of them to get extracted in this cooler environment. So that is the time where you'll actually have it in a vessel that can be chilled down, generally with glycol. There's jacketed tanks that have glycol around them. The grapes can be inside. They can be held at a lower temperature, and so you're able to just kind of prevent anything from happening for as long as you would like. Could be a couple days. It could be 7-10 days, like however long you want that cold soak to last. This is like a pre-fermentation step, after which time you would warm things back up and to start the fermentation process proper. Some things are done during cold soak. You can have you can certainly, this is the time I was mentioning before about the non-Saccharomyces yeasts. A lot of these non-Saccharomyces yeasts are active at lower temperatures than Saccharomyces. Saccharomyces cerevisiae being the main yeast that performs most alcoholic fermentation in wine. But there are other yeasts that do other things. They create other compounds that have been found to be really interesting. It kind of adds complexity and depth to wines, and in particular, a lot of these function better at cooler temperatures. So during cold soaks, people might add these non-Saccharomyces yeasts to kind of protect the juice, the must, during this process before the advent of Saccharomyces cerevisiae. And there's lots of different types of species, of genre and species of non-Saccharomyces yeast. An interesting thing about what we do here at Troon is like we have all of these things as well, right? So we've done some testing and looked at some of the different yeasts non-Saccharomyces yeast we have present. And so what we do here, as opposed to an actual cold soak, is we do like what I call an ambient soak. So our grapes are picked very cool, and we keep them outdoors. And going back to what I was saying about those cool nights, cool evenings, but then warm days, we can just choose how quickly we want to allow them to heat up by simply where we put the fermenter. Like if we put it in a nice sunny warm spot, it'll heat up a little faster. If we put it in a shady spot, it'll heat up a little cooler. And during this time, that's when our kind of spontaneous, but also I guess I will use the word native, non-Saccharomyces yeasts will start doing their activity. So that is something that we get for free here, but that is something that's commonly used by other winemakers who want to try to like mimic that process.
Yeah, so during this either cold soak or ambient period, for those trying to control and have a cold soak, just as a reminder, we mentioned a lot of places add a little bit of SO2 just to prevent anything else from growing during this time. But you typically are doing either a light punch down or pump over, or I've even done like bucket overs to be very gentle on certain vessels just because you don't want anything to grow on the top during this time. Again, this is focused more on extraction, tannin, other flavor components. But then as you move into the fermentation period, like Nate was saying, we use spontaneous fermentation here. All the yeasts come in on the grapes, but you do have the option to start your own. And I believe you have a protocol you talked about a little bit last time, a very specific protocol.
You just brought up something I wanted to touch on. You mentioned the enzymes and tannins. That would be something that would be added if in a more conventional approach around this time. Is there are lots of different enzymes that can assist with extraction. And so depending on what the winemaker is trying to get out of a certain type of grapes, they can add enzymes that will help basically break down certain components of that grape to make it more readily extractable, into solution. And similarly, tannins can be added, even during that cold soak period. And tannins can be added for structure, obviously, is how a lot of people think about tannins. They can also be added for color and color stability. And in addition, some tannins can be used for like there's a botrytis, so a certain type of fungal process that can come in on grapes. There's an enzyme released by the botrytis called lactase that can break down tannin, and as tannins. Additional tannins are sometimes added, they call them like sacrificial tannins, so that the botrytis the lactase will actually deactivate these sacrificial tannins, leaving the skin tannins of your grapes intact. So there's a couple different reasons people might want to add things at this stage, but then whenever you got what you wanted out of the cold soak, or in our case, once the ambient soaks warmed up, then the fermentation process begins, right? And for us, that fermentation process is spontaneous. They just threw out that kind of ambient soak. As it warms up, the yeast will start. We'll also get bacterial changes too. There are some bacteria that are also functioning at colder temperatures, and those kind of set the stage for a lot of the yeast, non-Saccharomyces and Saccharomyces yeasts that come later. But eventually, our fermenters all start fermenting spontaneously, whereas in many places this would be when the fermentation step occurred. And I think that's what you were prompting me, but now I've talked for a while, so you can talk about the fermentation.
Well, you had a great protocol. It's very specific, but I'm sure my explanation will pale in comparison. But essentially, you can pick out what yeast you would like to use for whatever wine you are creating. We kind of talked about this in the last podcast too. It's like there are so many now. We should have Googled how many exist, but you might have one that you always use for your Pinot Noir every year. You might also choose to do different trials. I've worked at places where we brought in, X amount of Chardonnay or Sauvignon Blanc, and they had 5 different yeasts for each that they were trialing that later we would do blends between those wines on. And so you can have some fun with it, or you might already know exactly what you want. But you essentially rehydrate this yeast, and you might even give it a little something to get going with, but you're trying to temper this solution to get it so that once you put it into the tank of juice, which you are probably heating up. I have seen people try more natural approaches to get the temperature up. And to be honest, I mean, yes, you want to already start heating up the tank, but if you put a bunch of yeast into grape juice, it will ferment. So you do want to kind of aim to create the perfect atmosphere for it to exist and not be producing any off flavors or, you know, not immediately take off. It's like you want to create the healthiest ferment possible. But once you go through your steps of hydrating this yeast, then you actually add it to the ferment. Depending on the size of the vessel too, they might have you do like a brief light punch down or a pump over just to kind of get it more homogenized in that vessel. And you can tell, so, you know, say you added a yeast to your tank of juice. If you leave for, you know, like 5 or 6 hours and you do something else and you come back and you try and smell the top of the tank, you shouldn't breathe in too deeply because it will already be producing a lot of CO2 if that worked well. It's same with spontaneous ferments here. It's like we'll leave one night and something will be in its ambient state, and then the next day we're like, oh wow, this really took off. So you can definitely tell when they've started. And again, you know, you can do a little research and pick the right yeast for your grapes. And so maybe you know exactly how something works and you know how fast it's going to take off and ferment. And that's kind of in your winemaking plan. On the other hand, if you're making more of a natural or low intervention wine, again, there are tools you can use to try and get it to do what you want. But you're not always exactly sure how fast it's going to ferment. We'll talk a little bit more about nutrients.
But yeah, with the yeast, I mean, I like thinking about yeast like breeds of dogs, because I think it's a good way of explaining like just that diversity. Because basically, like a purebred dog, right? It's a very known quantity. Like when you see the little puppy, like you know exactly what it's going to grow up into because you can see mom and dad and like and their parents and their grandparents. They're all the same breed of dogs. They can vary a little bit, right? You know, some are a little bigger, some are a little smaller, some are a little faster, a little slower, but they're all very, very similar. That's why they're a breed of dog. And like that's why a winemaker would choose one yeast over another, is because it would just have it's a very known quantity. It'll have very similar enzymatic activity that might release certain types of compounds. So it would prefer like certain flavor profiles. They'll ferment at a certain speed. Some yeasts are faster, some yeasts are slower. Like you'll know that. Same thing with Micah started talking about nutrients. Like some yeasts are good in very low nutrient environments. Some are a little more demanding and require higher levels of nutrients in that must to be successful, to be happy. So you can go through these yeast catalogs, and like Micah was saying, I don't even know how many commercial yeasts there are. I do find it interesting that I believe almost every single one of those were all isolated from a spontaneous ferment at some point. So that's kind of where they all came from. Is these aren't like for the most. I don't know that there are. I'm sure there are, but I don't know of like the genetically modified yeasts out there. I think most of them are kind of through quote conventional breeding, Okay, over 200 different. I bet, man, I'm kind of surprised it's only 200.
Yeah.
So, okay, quick Google search, over 200 different strains of commercial yeast available. But
we do trust Australia, so
yeah, they would know. But there's both. So Saccharomyces cerevisiae. There's also Saccharomyces bayanus, and there's so it's not all cerevisiae. There's different strains, different species of yeast that can do these fermentations. But yeah, after like Mike was saying, after the yeast rehydration step, you want to do that appropriately. If you're buying all this yeast, it would be pretty silly to kind of be careless with your inoculation and take all this expensive yeast and only like 5-10% of it survive your rehydration process. So if you are rehydrating yeast, please make sure you actually follow the instructions and Oftentimes this can be done with, it's not really a rehydration nutrient. Sometimes people call it rehydration nutrients, but if this rehydration is done in not just water, but in the presence of other compounds that basically help the yeast reform their cell walls after being dried out, that'll lead to a much more successful inoculation, as will that attemperation step that Micah mentioned. Like, when you're bringing the yeast kind of back to activity, they're going to be at a pretty high temperature, around 100 degrees or so. But then almost definitely your grapes, your juice, especially if you were doing a cold soak, is going to be significantly less than that, like 50 degrees or so. And so, you can't just dump 100 degree yeast into 50 degree grapes and presume that they're going to be okay with that. It's a pretty big temperature shock. So you can take steps to add juice back to the rehydrating yeast to slowly bring down the temperature until it's within a fairly close approximation of the tank temperature, and then you can add it. But speaking of adding things, though, this is other, and we'll talk about malolactic fermentation later, but there is the possibility of adding malolactic fermentation, so like bacteria, Enococcus, at the same time. Some winemakers, so this is called co-inoculation, and generally it's maybe 24 hours later, but in some locations they found it preferable to have your malolactic fermentation occur concurrently with your alcoholic fermentation, right? So the yeast that you just added to ferment the sugar into alcohol, you might also add this bacteria, Enococcus, it's not technically a fermentation, but to transform the malic acid into lactic acid. And we'll talk about that process later, but it is a possible addition at this point.
Yeah, so just to keep that fermentation happy, nutrients we didn't really make it its own step this time, but we discussed it last time. So you have some different options, and a lot of people do a combination of these if this is a more conventional style of making wine. If you are natural or low intervention, you're probably attempting not to do this or just kind of minimally using them, but...
Talk about yan, right? So one of the things to understand about your wine, whether you are inoculating with yeast or not, if you're doing spontaneous yeast, is how many nutrients, what the nutrients look like in that juice or in that must. And remember, we mentioned that generally 24 hours after fruit intake, you would be doing some testing to find out, certainly that's a good time to find out the sugar levels and the acidity of your incoming grapes. This is also when you would probably look at what's called YAN. It's YAN stands for yeast assimilable nitrogen, and that reading is a combination of basically two different forms of nitrogen present in the grape juice. There's basically an organic nitrogen, which is basically, sometimes it's just called amino acid nitrogen. And then there's inorganic nitrogen, which is ammonia or ammonium. And so those two forms of nitrogen are what's present, in the incoming grape juice. And that level is related to the vine nutrition. So the amount of nutrition that the vine has access to, if you were providing it, if you were spraying it, if you were putting it in drip, like fertigating with it, if you were like we do, we add compost in the fall. That compost breaks down, it adds additional, nutrients, nitrogen especially, back to the soil, and the grapevine picks it up. The yan level is related to, but not completely directly related to, the nutrient status of the vines. But anyways, understanding that yan level of the juice is important, because you'll want to know if there is enough nutrients present to be able to successfully ferment that sugar into alcohol. And the more sugar you start with in general, the more nutrients that need to be present in that juice or must to be able to successfully convert, for the yeast to be able to convert all of that sugar back into ethanol, which is of course what we're going for.
Yeah, so once you know those numbers, and if, so I will say with natural winemaking, obviously, you're going to want that yan to be exactly where you want it because you're not going to be able to adjust. If you are low intervention, you're attempting not to make any adds, but on a more conventional winemaking style, then you could be adding a combination of some of these things I'm going to list. So there is Fermade O, Fermade K, and DAP are the three most common things that different people add at different times, right? So it's like they might make a part of the add a little bit after that fermentation first starts, and then towards the end of fermentation add a little bit more. But basically Fermade well, and going back to what Nate said, there's different types of nitrogen too. So winemakers will kind of take that into consideration, but Fermade O replaces. Okay, so I'll start with Fermade K actually. So this contains some DAP, which is diammonium phosphate. You can do these adds together, or you can just do Fermade K. There's also Fermade O, which instead of DAP being in there, there's amino acids. So depending on your program, what you think is best, you'll be making these adjustments as you see needed. But with Fermade K, there'd be like a little bit more of a heat spike, and a lot of the times a little bit faster fermentation caused, because giving your ferment any amount of DAP is kind of like giving a kid candy. I think we brought this up in the last podcast. It's, not so nutrient dense, but it's going to give it, it's going to give it a little bit of activity. And so you can, again, use a combination of these things. You can just use one product or the other. But this is something that if you do need more nutrients, these are products you're going to be possibly adding. And then we did already kind of mention that some people will either bleed off, so it's called saignéing the wine, and then adding back water or tartaric acid towards the beginning to kind of adjust the chemistry of the wine. This might be another point during the fermentation that people double check that all of those numbers turned out correctly, especially at really big places. Sometimes people misread a work order, or you think that you had X amount in that tank and that you adjusted properly, but maybe you need to do a few little things to really get it to the right spot. So during the fermentation, you are monitoring daily, typically the Brix and pH. You might do like every other day at a certain point, especially if you have a little bit slower, steadier ferments, but there is a tracking that should be happening during this time, because if at any point your ferment is going to stall, you want to be able to catch that as soon as possible. And by that I mean, you're kind of watching the yeast ferment that sugar into alcohol. If all of a sudden the sugar is at about the same level for like 2 or 3 days, you can either find a creative solution to get it to keep going, or you might need to re-ferment, which in a lot of cases is extra time, it's extra product. So your goal is to get all of that sugar to ferment, not necessarily as quickly as possible, but you just want to make sure that that ferment is healthy and happy. It has everything it needs to completely finish, so that, as you're looking over your tank chart as a winemaker, say you have like 100 ferments going, every single time one finishes, that makes you a little less stressed. So you want to make sure that all of these are healthy and happy throughout the process. I don't know if we're going to go into stalled ferments a little bit, or
well, let's there's a couple things I want to touch on here, and going back to the nutrition for a second. So DAP, you mentioned it's diammonium phosphate, right? So that is the inorganic version of nitrogen that I talked about, that you would have those two components of yan, inorganic and organic. So DAP is the main method of providing that inorganic nitrogen. Whereas, so Fermato is a product name. There are multiple, different companies make different versions of this, but the O here is standing for organic. So that's the organic nitrogen version. And then the Fermade K that Micah was talking about is basically a blend of basically DAP, so the inorganic plus an organic nitrogen source. So there's a couple different ways of providing that nutrition to the ferments, and as Micah alluded to, the DAP is the inorganic method. It's actually very inexpensive, which is why it was even more so in the past commonly used. But I think it's getting less used because as we understand more about fermentation kinetics and the effects of DAP usage, as like Micah said, it's giving the candy to the kid. There's almost always temperature spikes, and then it can drop off really rapidly. They've actually shown that it has a pretty negative impact on aromatic, and in particular aromatic complexity of the finished wines, things that have had a lot of DAP in it. It can lead to reduction problems in the wines, so those kind of off odors like rotten egg kind of things going on. So it's not particularly great. And so at best, if you are in parts of the world where famously, Eastern Washington is one I know of that has just really low YAN levels. Like I've actually measured juice and gotten a non-detectable YAN in a grape juice before, which I didn't even think was possible. But that can happen in Eastern Washington. But either way, you can have very low YAN levels, and so like let's say like 30 YAN, whereas 150 YAN is maybe kind of like the minimum that people kind of think of in a conventional sense. It's what people might think of as like kind of the minimal acceptable, like safe fermentation. But again, like I mentioned, the higher the sugar levels, the more the YAN might be necessary. So you might need up to 200 YAN or something. Well, if you're down all the way at like 30 YAN, you would have to add something like DAP to be able to make up some of that difference, because you can only add a certain amount of organic nitrogen to the ferment, like legally, and anyways. But but the idea is to try to minimize DAP usage, certainly, and to preferentially use these other more organic forms of nitrogen. And the timing matters too. We didn't really talk about that, right? It's like you kind of said you use some of it at the beginning. Generally once you have an active ferment going, 1 to 2 bricks drop is when you might do your first nutrient add. And in particular, it's very important that be, if it can, that be the inorganic the organic nitrogen addition. This is kind of like priming the ferment for multiple generations. So it takes multiple generations of yeast to ferment wine. It's not the yeast you add if you're inoculating. Those yeasts that you're adding are not the ones that finish the ferment. It'll be the offspring of those yeast, 6-7 generations maybe that finish the ferment. And by adding inorganic nitrogen at the offset, you're kind of creating a situation where those successive generations of yeast have problems utilizing organic nitrogen well. Whereas if you provide organic nitrogen, there's going to be kind of these transport proteins that assist with transferring the organic nitrogen into the cell wall. Like those proteins will be strengthened in those initial yeasts, and that kind of sets up subsequent generations for success in utilizing this like better form of nitrogen. So anyway, a lot to say about that. But then we were talking kind of about the fermentation, but we didn't really go into the cap management too. I don't know if that's something you want to talk about. Like, you kind of talked about taking the readings, right? Like probably daily temperature and readings, but like what we do during fermentation itself.
Yeah, so there are a lot of different options, and you could use many different size fermentation vessels. So just to kind of visualize, it's like you could use a barrel, which we use a lot of barrels here at Troon, but then you also could be at a facility that has like a 4,000 gallon tank that you're fermenting in. So there are a lot of different scales, and just to kind of understand, if something is that large, you're going to have to do calculations on how long of a pump over you're going to want to do on a tank like that, versus if you're fermenting in a bin or a much smaller tank. Like I've seen, the 1 ton tanks are very popular, or like fooders even, it's possible to do punch downs on. That's something where you can actually fully submerge the cap using a punch down tool, or even your feet or your arms, right? So, depending on how much extraction you want, and what exactly you're going for in your winemaking program, you can be punching down, you can be, like I mentioned, using your hands and being like really gentle. You can be pumping over, you can be doing a combination of these, but essentially you're submerging the cap, because we're talking about red wine specifically today. So that could be all destemmed fruit, it could be whole cluster, it could be a combination. Obviously, when whole cluster goes in to start, if you're using a higher percentage of whole cluster, those punch downs typically are a little bit harder to first start breaking through. But as you go, the cap is going to be a little bit easier to sink. And it's also a really good method to kind of pair managing your cap with taking your Brix and temp, because then each day when you come in, say this fermentation was sitting for the last 12 hours or more, you do your best job of mixing everything and punching down the cap, and then you're going to get a little more accurate reading. Whereas if you just immediately take a sample off of the top, or X amount of wine off the top, it's going to be a little bit hotter, and possibly you're going to get different numbers from that part of the bin versus making sure it all fully gets mixed together. I don't know if there's anything else you want to add to cap management.
I was just, we these podcasts go out to such a diverse audience. I was just going to mention what a cap is.
Yeah.
When you're with white wine and with rosés, you're just fermenting juice, and so the juice, that's a more straightforward fermentation. With red wines, and technically this is red winemaking, but the same would also apply to orange wines or amber wines. Basically, where you're fermenting white grapes in the same way that you'd ferment red grapes. Anytime you actually have the grapes and or the stems, when during fermentation, it forms a cap. Once fermentation is initiated, basically the CO2 gas is being given off, and each of those little grapes kind of becomes a little like reverse parachute, and the CO2 like rising up kind of pushes all those skins and everything up to the surface, where they kind of float on the surface of the liquid of the fermenting wine. And the idea is that we've talked a little bit about it right now, but a lot of the really interesting stuff that makes red wine and amber wines, into what they are, comes out of the skins of the grapes. So a lot of the tannins, the color, interesting flavor compounds, aroma compounds, like a lot of those are found in the skins. And so if you did nothing, the skins would be floating up on top of the wine, where they would just not really be touching the fermenting wine as much. So there's different methods that you would use which we just collectively call cap management, like ways of getting that cap like resubmerged into the fermenting wine, so that you can continue to have that contact throughout the fermentation.
And I'll also mention, if you're making a natural or low intervention wine where you're not using sulfur, it's especially important, because it's like you don't want anything to grow on the top. If something is growing on the top, though, what's the best way to manage that?
Oh, well, yeah. Well, we attempt to not have that happen. And the way that we do that at Troon, and this isn't really an additive, but I guess I'll talk about it, is to all of our fermenters, we add dry ice. And so this is more, I guess we should have talked about this during that kind of ambient soak portion. But, dry ice, a lot of the things that grow that you don't want growing in wine, is directly related to the amount of oxygen exposure or not that is happening in whatever vessel it is. So in the fermenter, yes, but also later on in the aging process in barrels. When you have things, a lot of spoilage bacteria or just bacteria and or yeast that have negative components to them occur in the presence of oxygen. And so if you can minimize that, you can really prevent a lot of problems from happening in the first place. And so we add dry ice twice a day to the tops of the fermentation vessels as they're kind of coming up to temperature, as they're warming up. And what that does is that just minimizes the amount of air, the amount of oxygen that is at the surface. And that's where a lot of these things, might grow that could provide off flavor to the wine. So if you can do that for the first, several days while that fermentation is beginning, then when the fermentation starts and the fermentation gets really active, that's when you will get a lot of CO2 being given off by the yeast itself, right? So during fermentation, as the sugar is converted into alcohol, there's also CO2 and heat that's given off. So it warms up and it gives off CO2. So when that happens, it basically starts protecting itself because the fermentative yeast themselves are pretty good at like these low, anoxic type conditions. Like they prefer some oxygen present, and that's one reason why you might want to do a pump over, for example, that splashes the juice a bit. It adds a little more oxygen into the fermentation once you have that active fermentation. But you wouldn't really want to add oxygen beforehand because you could be leading to the growth of some of these other spoilage organisms. So we try to prevent the growth through the use of dry ice. So just keeping those under a blanket of CO2 up until it basically blankets itself with CO2 through the active fermentation.
Yeah, and if you ever found yourself in a position where you do smell something a little bit off, I was trying to set you up for saying that it's actually better not to punch down or pump over the cap, so all the grape skins in that case. I've been at places where somebody smells the fermentation, they're like, oh, this, something's wrong with this. And their immediate reaction is like, okay, let's do a punch down on it. But really, you're just possibly continuing the growth of whatever was on top. So there are ways to stop that. Like, you could spray SO2 on the top of it. You could attempt to get some of the grapes off the top if this is obviously in the case that you can actually touch it. Like, you wouldn't try to do that in a giant tank. But, this is just going back to why it's so important to actually be managing the caps and making sure that no fermentation vessels get skipped. Before you leave for the night as a winemaker, you typically have a list, if you are using glycol, where you'll have someone walk around and they'll check all the temperatures of all the tanks. Because yes, you could have taken the bricks and temp and done all of the work orders properly during the day, but say you leave and something's not set at the proper temperature, then you can completely ruin all the work that you've done on a cold soak or a ferment.
Yeah, actually that's a good point. We should talk about controlling temperatures. So controlling temperature during fermentation is definitely a thing. And depending on the winery, it could be common that they would use this to their advantage by, say, having much warmer temperatures like right at the start. Especially it will help if they've just inoculated. They can have that. They can warm up the tank. We talked about how the yeast, when you're rehydrating them, are pretty warm. They warm up the tank, add the yeast there, and if you want something really warm, it's kind of nice to have it on the front end. If you're controlling that, because the type of extraction that you can get before there's a lot of alcohol present is generally speaking the kind that you want. And then they might be controlling that temperature to fall to slightly lower temperatures later on in the ferment, and then kind of getting cooler towards the end of the ferment. And that's when you have the alcohol present and you might start extracting more bitter tannin compounds, particularly as the grapes have been falling apart. The seeds are now being exposed to the fermentation. Seed tannins are shorter tannins. They're more bitter tannin than the skin tannins. So you might want to minimize the extraction there. What we do at Troon, we don't use temperature control per se. We do use forklifts though. So we mentioned that our fermentations are outdoors, and we can to some degree use where we position those fermenters. In the sunshine, like I'd said before, in the cold soak, you can either keep them in the sunshine during fermentation if you want them to keep warming up, or you can put them in the shade. And there are these oak trees we have back there to kind of keep them cooler, particular towards like once the fermentation is pretty happily started and going along. We do move most of the things over into that cooler area just to keep the fermentation temperatures cooler, which also means a longer fermentation. So in general, the warmer the fermentation is, the faster it's going to be done, and the cooler it is, the longer it will be. So it's and different compounds, different tannins, different structural elements of the wine are extracted at different temperatures at different times of that fermentation, and so trying to line up those temperatures with what's happening in the fermentation and what you want to be happening is kind of part of that options that winemakers do. Many wineries have temperature control. Each vessel has its own temperature control. You can just literally dial in the temperatures you want when. And that's what Micah was talking about. Going through and checking all the temperatures, making sure they are what they're supposed to be. We do take temperature readings every day, along with the sugar level readings to track the fermentation. And that helps guide us in our process here at Troon of just deciding if we want to forklift the fermenter back into the shade or if we want to keep it in the sun. So temperatures are definitely an important consideration during that fermentation process. There's different options for controlling that.
Yeah. So then do we maybe get into pressing and settling?
A little bit about stuck fermentations maybe?
Sure, yeah. So one reason you really want to avoid mismanaging your fermentations is that if you do see that something has the same amount of sugar, and a lot of the time that goes in hand with the fact that maybe the temperature is not quite where you want it, you can, number one, fix that, but also it might be to the point where you're like, okay, I need to essentially restart this or quickly kind of try to fix it. Like, you don't want to leave it just sitting for too many days because that could produce off flavors and just completely stop it. So if the population is maybe dying a little bit, if you have 2 or 3 tanks of something that's very similar sitting next to each other, you could say, okay, tank 6, 7, and 8 are all doing about the same thing. It's just that tank 8 stalled a little bit. So maybe you choose 6 or 7 to do a little bit of a swap, or just take some out of the fermentation that's going properly, put it into the one that looks like it's about to get stuck. And so you don't even have to rehydrate any yeast, adjust the sugar, use any type of product. But sadly, sometimes if you wait a little bit too long, or for whatever reason you've tried to add some of the fermentation from another vessel to get that one that's stuck going, if that's not working, what that can cause is that you'll need to do another fermentation, and you'll need to even add sugar sometimes to get it up to a certain Brix so that you can actually re-ferment and get that going. So obviously this is not only X amount of time, and it's a little bit stressful depending on, the size of the vessel. Maybe it's like a single lot wine or something that, you really want it to turn out a certain way. Well, if you're adding all of this extra product, it might kind of change the flavor. You're also using more product. So again, it's just more expensive. It's a little more time consuming. So your goal always is to set up a program for yourself and a lot of checks and balances so that you can just go through a fermentation and get straight to that pressing and malolactic fermentation, and you don't even have to stress about, analyzing a ferment that you think is dying or that you come in and you smell and you need to restart.
All right, so now let's presume that we've just gone through the fermentation process, everything was successful, didn't have any stock fermentations, whatever. We're getting down to the tail end of the fermentation, and this is when we start thinking about when we want to press this off, right? So obviously the red wine, the orange wines, they're fermenting on their skins, sometimes on their stems as well. And so when do you put that into the press? When do you separate that from the skins and the stems, and go to liquid only? So that decision, like when to press that, there's somewhat stylistic implications for like why you might choose when to press it. And in general, I guess you could kind of say there's like fairly simply there's 3 times, right? Do you press it off like at dryness, meaning do you press it off once all of the sugar is gone, it's all been converted into alcohol and the fermentation is basically done? Or do you press it off slightly before dryness, or even like significantly before dryness? So you press it off, and after you press it, it will continue to ferment because there's still sugar left. Or do you do what's called an extended maceration? Do you leave it sit on the skins for some period of time? And there are some extended macerations can be like very long, like months. But it's more commonly maybe is a couple weeks even of extended maceration where the fermentation's done, but for one reason or another, you've decided to let the wine just basically soak on those stems and skins for some period of time after, and then you make that decision when to press. And so, there's different reasons why you would do these. I guess
it's a bit of the fun part too, because like for us here, we have X amount of bins outside specifically for our red wine. We also use amphora and concrete, but for all of these vessels, it's like we hit a certain number and we know that we're going to be pressing that's coming up, and so we'll add another few vessels every day during fermentation to our tasting plan for the day. And so we'll have notes daily and kind of decide like, okay, we want to press this one tomorrow, or we want to wait a few days. We at Troon are basing it, again, as everything, on numbers and taste. There are places that don't always have the same luxuries, and sometimes if you have a really large facility, there are X amount of tanks, and when you're pressing off skins, that means that you're removing all the wine at this point, and you're putting that into another vessel, and then you're pressing all of the skins and possibly clusters if you did whole cluster, and that's probably going into another tank because you'll have different press fractions, which we'll talk about. But that means that you need other vessels because this wine is coming out of one and into other ones. And so sometimes it's not just about taste, it's also like, what do we have available when? And you're kind of trying to like puzzle piece together how it's going to work. But we are typically pretty lucky to where there's not really a limit of what we can do when. There is for our press, I would say, but it's like things come in typically in a way where we have a little bit of a window. So we get to decide, when things taste the best, and once numbers and taste are right on point, then we continue to the pressing step.
Yeah, that larger facilities, the whole kind of like Tetris of trying to fit everything that you have in is very real. So a lot of those pressing decisions can absolutely be made on like, we need to get this empty, so we're pressing it now because then it can go into like barrels or something that's like not eating up our tank space, and like we absolutely need this tank space. I mean, it's pretty typical. One of the wineries I worked at, we had to flip tanks, at least 3, if not 4 times during harvest, right? Like you would have to use the same vessel for fermentation multiple times just to make the winery work. Like you didn't have enough vessels to ferment 100%. of what you have. You only had enough vessels to ferment like a third of what you were actually bringing in. So you had to have those early ferments, start, get done, and get pressed off so that tank's available for the next thing to come in. So pressing decisions can absolutely be made for that reason.
It's kind of like, it's funny you said that, because it's like flipping tables during a dinner service, right? Totally. It's like, you have to get X amount done. And that also kind of going back to the fact that we were talking about picking different yeast strains and different protocols for making your wine and for fermentation, that can also be due to this, just being like, okay, we're going to make X amount of wine this year and these need to ferment quicker, right? So it just totally depends on, size of your operation and how many people you have helping and the style of wine that you're making.
But at Troon in general, we pretty much always press off like at or before dryness here. So the before dryness, so our glue, our carbonic is pressed significantly before dryness. It's pressed off, following the carbonic maceration, which can have like 50% of the sugar or more available. But like different varieties, like Micah said, we go through and we start adding things on as things are getting ticking down, lower sugar levels. Certain grapes, especially that have certain tannin profiles, you might want to press off earlier. We used to grow Tempranillo here, for example. We no longer do, but that's a popular variety here in Southern Oregon as well. And in Oregon, Tempranillo can just have these massive, like monolithic tannins to them. And that might be a reason why you might want to press off at like 5 or 6 bricks. You've already extracted plenty of tannin. You don't need more. And so you might press that off a little earlier. But in general, most of our wines are pressed off right around dryness level, but it is based on us tasting each of those fermenters. We've pressed off on a fermenter by fermenter basis. We don't press off by lots. That's really useful for us too, because all of our fermenters are often a little bit different. Like going back to that whole cluster, we might have one fermenter. So like when the grapes come in, it's the same lot of fruit, right? But one fermenter might be 100% destemmed. One fermenter might be 50% whole cluster. One might be 75% whole cluster. So they're all fermenting right next to each other, but they're pretty different wines because of those different conditions. And so we'll want to press those off separately and make different press decisions on those. But as we load them into the press, actually the first thing you do is you separate out the free run juice. So you want to drain off as much wine basically from the skins as possible before you load it into the press, and that's generally what's known as the free run. And then as you load the press, you start pressing. As the pressure intensifies on the press, it goes through a press cycle where it'll inflate, deflate, maybe roll around a bit to try to get the grapes shuffled a little bit, then press again a little stronger this time. As you're doing that, you're both looking at the pressure and tasting it, and that's when, like Micah said, you'll make some press cuts. You might have a light press cut and then a hard press cut at some point. I wanted to bring up the hard press in particular because at a lot of wineries it might be common to treat the hard press wine separately. At Troon we do make multiple press cuts, and we actually will separate them, settle them separately, and barrel them down separately, and taste them separately, make different blending decisions on them separately. But at other facilities, you might, for example, add additional enzymes to your hard press fraction. Hard press generally has a lot higher turbidity. It has more suspended solids in it, and you might make the decision to add some enzymes to help clarify that hard press before it even goes into ultimately probably a barrel, but into whatever aging vessel it is. We don't do that at Troon, but that's yet another reason to make some of these press cuts, is that you can treat them differently in the winery.
Yeah, and just to explain that a little more, so Nate mentioned there's the free run, which is where you're just draining that fermentation. And to kind of give you a visual, it's like free run could go into tank 1, and then you collect all of those grape skins and possibly whole cluster as well. You press them, and you have a light press that then goes into tank 2, and then you have a hard press that goes into tank 3, and from there you settle them. You're going to rack all of the wine off of the lees and into a vessel. So what could happen if you're going to put all of that into barrels is that you have your tank 1, your tank 2, and your tank 3, and they essentially were all the same grape from the same vineyard, but then they go into separate lots to continue their aging period. And we do that here. We keep a lot of things separate because it's kind of fun for blending purposes. There are definitely taste differences, and the chemistry is a little bit different. There are also places, though, that put everything into one tank, or you also could just have such a small fermentation, a small amount of one lot of one grape that it doesn't really make sense for you to separate it. But again, another stylistic choice, I would say, is that you could kind of divvy things up. You could even put some of it into barrels, some of it into amphora, which we've done too. So this is another step where you get to kind of play as a winemaker and do some trials, or just, create a more interesting wine with a lot of different vessels.
And sometimes before you get to that step you were alluding to, like racking it into an aging vessel, you, depending on if you're going to inoculate for malolactic fermentation, you might do it like in that settling vessel. So Micah's tank 1, 2, 3 analogy. If you're going to add bacteria, so we mentioned this at the outset. So fermentation, the alcoholic fermentation is with yeast, and that's what's converting the sugar into the alcohol. But then malolactic, it's still called a malolactic fermentation, but MLF, but it's performed not by a yeast but by a bacteria, specifically almost always Enococcus, although there are other bacteria that can do similar things. And this is converting malic acid in the grape into lactic acid. If you're going to this process happens spontaneously. It happens spontaneously at Troon, and it happens. This one is, I would say, kind of maybe 50-50ish or whatever. If people inoculate, this is maybe my Oregonian perspective. I mean, a lot of the Pinot Noir, Willamette Valley Pinot Noir, a lot of that is spontaneous malolactic. But that's because Pinot has like better acidity and things.
I'd say that's true though, because there have been places that I've used a lot of other products, but then we get to that stage and it just spontaneously occurs. So yeah, it's probably.
But if you were going to inoculate it, you'd probably want to do it when it's all in one vessel, because as we were talking about this earlier, and if you don't do it, then you can do it barrel by barrel, but you're basically taking like teaspoons of liquid, and like what if you have hundreds of barrels? So like you don't want to have to inoculate hundreds of barrels.
It's a great way to kill a few days.
Yeah, malolactic. So before you rack off, those tanks, those settling tanks, 1, 2, 3, like Micah was talking about, you might add malolactic bacteria here, and if you were, one of the benefits, I guess, of inoculating for malolactic is that just like yeast, there aren't as many strains of malolactic bacteria that you can purchase, but there are quite a few, and different strains, just like the different breeds of dogs, they're better at different things. So some are better at like low temperature. For example, if you have a very cool cellar, the malolactic does better in like the 60s, mid 60s even degrees in the cellar. That's when malolactic prefers to happen. But if you have a really cold cellar, like we do at Troon for most of the winter, then malolactic is pretty slow. So some strains are good at low temperatures. Some strains are better at in terms of the acidity, the more acidic the wine is, the more difficult it is to go through malolactic. So there are some strains that are good at higher acid, lower pH settings. High alcohol is a problem. It can be a problem. So there are some strains that are better at high alcohol wines, of getting them to go through malolactic. And then sulfur sensitivity. So generally, most bacteria are rather sensitive to sulfur. It's like why sulfur is added in the winemaking process many times is to help control bacterial growth. And it has antioxidant capacity as well, preventing oxidation, but the bacterial control is one of it. And like I just said, malolactic fermentation is done by a bacteria. So if you've already added multiple, maybe once or maybe even multiple times, if you've already added sulfur to your wine, like at crush, maybe during the cold soak or something, there are certain strains of malolactic bacteria that do better if there is a decent amount of sulfur present, whereas normally that might completely inhibit malolactic bacteria from functioning.
Yeah, and so we kind of mentioned a few of these steps, depending on what your program is, they could shift a bit. But just as a reminder, I mentioned when you're pressing, you press into a vessel and you would free run into a vessel, and then you're racking off the lees and into vessel, whether that means that you've already added a malolactic bacteria or if you're doing it vessel by vessel after. But we go into this period where some people, depending on their wine program and what variety they're working with, they might even rack off lees again at some point. But you're kind of going into more of vessel management and the aging process after the malolactic fermentation has completed. So you really want fermentation and malolactic fermentation to be complete so that you can start this period of aging and management. And so you can decide how you want to do this, but most places I've been at, let's just use barrels for reference right now. It's like if you want to send any type of samples into a lab or do anything in your lab, you would probably do that step first. Then you would be possibly testing for SO2 in addition to some other things. And by that, I mean how much free SO2 and maybe even total SO2 is present. Then you would make work orders, or, maybe you're in a small winery, but you would do your SO2 adds to follow, and then you would top the barrel. And you kind of create this cycle where you're checking to make sure that your sulfur management is properly getting done. And then you're also topping your barrel to keep it safe. So some places do not add any sulfur, so you wouldn't be doing this, but they probably should be still checking the chemistry every so often. You do typically want to top your vessels, or if you have like a variable capacity tank, you want to maintain that seal is actually sealed, or if you need to be gassing headspace, you can do that. But you'll create some sort of program where you're kind of checking on your wine, maybe like monthly or every 6 weeks. Sometimes here, though, we even do that every 2 weeks just to make sure everything's protected. But this period just ensures that you're maintaining your wine. And I guess we also kind of wanted to talk about, before you even put wine into a vessel, maintaining the vessels themselves. So you have a lot of options, but I'll just go over barrels really quickly. So here, for example, at Troon, we use used oak only. We source them from places we trust. We know they're well kept, and then we also look at them before actually accepting them. We know that they're cleaned and steamed, and then we get them here to our site, and we have another process where we just steam them, sulfur them again for storage, and then we maintain our barrels monthly, or about every 6 weeks. And so we make sure that they stay hydrated, and then also SO2 them regularly.
This is when they're empty.
Yes.
Yeah.
Yeah. So no matter what the vessel, you want to make sure that prior to putting anything into it, it's clean and hydrated and ready to be able to keep wine in it safely.
Yeah, so we kind of jumped to a couple different phases there. We were talking about malolactic fermentation. Sometimes that is added, to that vessel where you pressed off into, that settling vessel. but a lot of people don't even add it, right? And so they're going to rack off of those settling vessels directly to whatever is going to be this aging vessel. And Micah was using the example of barrels. Barrels, of course, for red wines and orange wines, it's a very common vessel to use. And I did want to talk about oak barrels specifically for a little bit, just because this whole podcast is kind of talking about different additives that might be used in the winemaking process, and I absolutely think we need to be talking about oak as an additive, because often a lot of wineries think almost like the more the better, right? And some people have even almost trained their palates to believe that the taste of oak means that it's a good wine, just because oak barrels are really expensive, and if you use a lot of expensive oak barrels, you're going to charge a lot for your bottle of wine to pay for those expensive oak barrels. And so when people drink like expensive bottles of wine, sometimes they're really oaky, and people are like, oh, that's what really good wine tastes like. But that is often because oak can really dominate flavor profiles of wine, sometimes to the extent that it's almost like obliterates varietal character. Like you can't even tell like what grape variety that came from. And this is how the extent that which oak really is used as an additive. Yeah, oak barrels serve a purpose, right? They're holding the wine for aging, but you don't have to get oak from oak barrels. Like a lot of people who don't want to have to buy those really expensive oak barrels, they'll age their wine in stainless steel tank, but then they'll add things to the tank that basically add oak flavor. Kind of best case scenario, I guess, this might be like just hanging oak staves like down the middle of that tank, so that the wine is in contact with those oak staves. So kind of the opposite of a barrel, right? Like in this case, the oak is in the middle rather than the oak being on the outside, kind of holding in the wine. It's the oak floating in the wine. But there's also like chips, little oak chips, or little like oak cubes, little squares that can be added. Sometimes people will add these to neutral oak barrels to make them taste like newer barrels, but like there's like oak dust, but there's even liquid oak extract, and this is like where you just have to see this for what it is like you can actually just dump liquid oak into a wine, and it makes it taste like it has been in like a brand new barrel, right?
You can put it in there a week before bottling.
Yeah,
if you
like
want.
you can do blending trials of like how much oak you want in your wine, and you're like, oh great, like we'll just add a couple gallons of this, and then like there you go. And this isn't, these aren't ever disclosed on labels. Like none of these things are additives that like are listed. So I don't know. It just really bothers me that people use that. What I really like, and this is going off on a tangent, but so all of our wines are organic, but they're also Demeter biodynamic, and we have different standards for our vineyard practices for Demeter, for biodynamic in both the vineyard and in the winery, and like right at the beginning of the winemaking standard for Demeter, or it's not right at the beginning, but it talks about the use of oak in winemaking, and it says that the use of oak cannot like fundamentally change the wine. Like Demeter wines should express like the typicity of the wine, and the oak should not get in the way of that. I mean, they are vague about it, but at least they do say like right there, they do not want oak to be a like overly dramatic influence on the flavor of that wine. So that is in a nutshell why we use all neutral oak barrels when we do use oak barrels. Like Micah said, we maintain them well, they're clean, and we use them. They're a good vessel to hold our wine in as it ages, but it's not imparting really any flavor. But then, of course, we also have like concrete tanks, both for fermentation and for aging here. And we have amphora, a couple different sizes of amphora. We've gone into an entire podcast talking about alternative vessels, so I won't do it now, but I really just had to mention the oak thing because, I mean, especially when you can just dump in liquid flavor, you're adding it just to change the flavor of your wine into something that never came from a grape. It's an oak tree, it has nothing from the vine. So if people want tannin, that's the other thing oak provides more tannin and structure to a wine, which is what a lot of people talk about. You know what? Whole cluster does that as well. and that is part of the grapevine. So that's what we do at Troon. Is if we're looking for the additional structure that some winemakers will try to use new oak to get additional tannin structure. Only their tannin is coming from oak barrels. We can get tannin from whole cluster fermentation and from other things that add that structure. So, all right, stepping off the soapbox. Let's move on in the process.
Yeah, so we're now to this aging process, like I mentioned. In addition to making sure that if you're using sulfur, you're maintaining the free SO2 you want, and topping your barrels. There's also just making sure that, the entire process of making wine, you should be paying attention to your cleanliness, because you don't want anything else growing in your wine or your vessels, right? So, a huge one is Brett. As you're topping, as you're running numbers in either your lab or sending samples to a lab, these are things that winemakers worry about, that one in particular. But throughout this process of aging, that kind of runs into the blending process. I don't know if there's other stuff about aging you want to cover.
Yeah, we've got other things.
Get into
so we kind of have these different stages, but like there's no fine lines or solid lines between them. Basically aging starts as soon as malolactic finishes in some ways, but of course the wine's aging during the malolactic fermentation as well. And just to be clear, Mike was talking about the need to top up and add SO2 and things like that. The barrels, especially if you're in barrels, not like a variable capacity tank, like Micah mentioned, like you wouldn't need to top that. A variable capacity tank has a lid that floats on top of the surface of the wine, so it basically it's always full. The tank is always full. There's no headspace there. But wines and other porous objects like concrete and amphora are also porous. Eventually through evaporation, they'll lose some volume of wine, and they need to be topped to be kept full. Going back to what our discussion of adding dry ice to fermentation, when we were talking about a lot of spoilage organisms need oxygen. Well, if you have headspace, that provides a space for oxygen, and that's when you'll be getting spoilage organisms or other organisms growing that you don't want in your wine. So keeping those vessels topped up is important. This needs to happen during malolactic fermentation as well, especially at Troon. Our malolactics can take months and months to finish. Back in the day, like it used to be nice having all of the fermentations were done, all the malolactic fermentations were complete like before Christmas. Everything got buttoned up and everything was done. It's
Which
mid-May,
is also
and we're
one
still
reason
not done.
for ads too. Sometimes
Yeah.
it's like people don't want to stress over waiting. So obviously with natural and low intervention wines, it's a little bit more stressful probably.
Yes, for sure. But the nice thing is if you do have patience, these processes generally all occur on their own. It's just a matter of time frequently. But so once malolactic finishes, that's when you would do those first SO2 additions into the barrels. I don't know that we've specifically talked about that at Troon. So, like we do this on a barrel by barrel basis. So even if you've got 10 barrels that, Micah's tanks 1, 2, and 3, all of tank 1 goes into like 10 barrels. So it's all basically the same thing, the same wine, but all of those may go through malolactic at a slightly different rate. Each barrel, each vessel becomes its own little universe. It does its own thing slightly. And so we treat each vessel separately. And that includes determining when malolactic is truly done. You can look at numbers for when this happens. You can be tracking like how much malic acid is present and how much lactic acid is being created. We use paper chromatography for this. It's an easy way of visually seeing how much malic versus lactic acid is in each barrel. But then ultimately we can just start testing the malic acid directly and seeing how much malic acid is present. When all of that malic acid is gone, we know that MLF is totally finished, and that's when we can consider adding sulfur. That's the first time Troon would add sulfur, is when that's finished. But we would only do that after tasting each barrel to make sure that that the malolactic is truly not just done like on paper, like, you know, it's not, that's just non-detectable malic, but that it also tastes complete. Malic acid has a very green apple-y flavor to it, and then during malolactic fermentation, there's a lot of like yogurty type character that occurs, and we want to make sure that all of those green apple and yogurt flavors are no longer present in the wine, and that's when we know that malolactic is truly finished.
Yeah, and so Nate mentioned we use chromatography to track this. We do also follow that a few weeks after it looks like it's finished on chromatography. We use Essentia to run a malic test, and you could either do this in-house like we do. A lot of people choose to send a sample to ETS or another lab that they trust just to really verify that it's done, because as I mentioned before, you want to make sure that fermentation and malolactic fermentation are complete before you would be adding SO2 if that is in your program, and continuing into that monitoring of each vessel with possibly SO2, and then also topping to prevent against oxygen.
Yeah, so once that's all done and you're on that aging process, and this is where Micah mentioned, you know, you might you might rack the wine off of those lees, and in particular, you might do this multiple times. Some people might wait until malolactic is finished. They might rack the wine off of the lees. They've added SO2, which helps, like I said, it also has an antioxidant capacities, and so the wine's a little bit more protected now when you rack it out of the barrel, like into a tank, for example, and then you either clean the same barrels and then remove the lees from them and then rack it right back into those same barrels. That's like a rack and return. If that's what you're doing, oftentimes people may add more lees at this process. You can, like many additives, you can buy things that are naturally there, you can pay money for. And so there's different kind of yeast derivatives. You can buy basically lees in a bag, and you've racked off your lees, but you can add these other lees back into the barrels. Lees, of course, are generally the dead yeast cells from left over from the fermentation, mostly, and over time during the aging process, those break down. They actually help protect the wine during aging as well. They do have antioxidant capacity, but they also start releasing interesting compounds into the wine during the aging process. Manoproteins, other interesting things that help with the texture of the wine and just the ageability of the wine. So if you are getting rid of your lees, sometimes people might choose to add more back.
Yeah, I'm glad actually you also brought up racking things into tank and then returning, because that's also a great checkpoint. Everything from one lot is in the same tank, so you could essentially just do an SO2 add in tank and then return to barrel, or also get a more accurate reading to send to a lab if that's something that you do periodically. I guess we could also talk about Brett. Right. And some different products.
Well, let's talk about the free sulfur levels, since you just
Sure.
brought up sulfur again, and the maintenance of those.
Yeah. So we've been talking a little bit about free and total SO2. So if you are using SO2 in your winemaking program, there are typically kind of target points that a winemaker will decide on for white wine, rosés, and reds. And so there is a legal limit to how much SO2 you can put in your wine. Also, your goal typically is to use as little as possible. But when you start using it, it kind of just starts this cycle. And so you don't want to add too much. Like when you're first learning about sulfur, it's like, oh, well, why couldn't I just put X amount in? Because that's how much I'm going to need, right? But your total sulfur that's going to show up when you're running numbers, that's the total amount that has been added to that wine and shows up on your test, whereas free is what's actually available to be used. And so you're kind of learning what that wine needs and wants. So it's similar to talking about tracking malolactic fermentation and how even if the same wine goes into 10 different barrels and it's all the same lot, right? It was all the same wine fermented and pressed the same way. Well, you might think, oh, okay, all of these are going to be going through malolactic fermentation the same exact way, but they're not. And so it's same with adding sulfur. You could be taking a sample of all of these 10 or 20 barrels from a lot and getting one number back, but they all could be slightly different. So you're trying whatever is in your program. Sometimes, again, it's testing lot by lot at a larger facility. At a smaller place, it's typically vessel by vessel because you want to make sure if you don't have to be adding extra sulfur, you don't. But you'll run tests periodically. Again, I think the most common that I've seen is like every 6 weeks you'll take a reading and
Of sulfur levels.
That
you're trying to maintain, yeah.
Yeah, so again, initially you might add your first addition of SO2 right after malolactic fermentation is complete, and then you'll take a reading. I like to wait at least like a week typically just to make sure you actually get an accurate reading on what's stuck. But from there, you don't really need to know exactly what the total is until a little bit later, but you'll just be tracking either, in Innovent on your spreadsheets, how much you've added so but you're trying to maintain your free sulfur just to make sure that it's at the correct level to actually protect that wine. And then that's in combination with the fact that you're going to be topping that vessel. So I mentioned earlier, a really good way to do it is if you want to know what the free sulfur is / if you want a sample of wine for anything, so like a blending session or to send to ETS for alcohol or whatever it may be, you would typically take wine out first to run that sample, want to run the test from that sample, then you would get your results back. You would add whatever amount of sulfur you deem appropriate, and then you would top the barrel after, because that way you're not topping and then needing to take out wine to put more sulfur in. It's just kind of a nice way, like routine to get in. And if you do a lot of blending sessions too later on, it's perfect because it's like, even though you're taking things out of whatever vessel it is, then you're consistently kind of following behind and making sure that numbers are correct and that it's, again, protected from oxygen.
Yeah, so, in a more conventional approach, like Micah was describing, you'll have a certain free sulfur level that you're trying to basically maintain throughout the entire aging process of that wine in the cellar. Let's just say that's like 30 parts per million or something like that. That could just be something you're trying to keep because that with that much free sulfur present, free sulfur is basically the percentage of sulfur that's available to both help control oxidation. It can be antioxidative, so help kind of soaking up the byproducts really of oxidation, aldehydes in particular, binds with those, as well as there's an antimicrobial perspective of that, which is related to the pH of the wine. The more acidic the wine, the lower the pH, the less sulfur is needed to effectively combat microbial spoilage. So a lot of, like Micah said, roughly every 6 weeks, they might, add a little bit more sulfur just to hit that like 30 part per million, target level of where they want to keep their wines. That is not what we do here. We do, because like Micah said, once you start adding sulfur, you end up like always adding sulfur. And so while we do add sulfur, it is the only additive that we actually do use at Troon. we use very small amounts. So there's, wines in the United States legally can have no more than, pardon me, can have no more than 350 parts per million of total sulfur, and whereas biodynamic or organic wines can have 100 parts per million of total sulfur, of which only some fraction of that is free, right? Troon wines generally have 30 to 40 parts total sulfur. And so, we have very low sulfur levels. If we tried to maintain a 30-part free sulfur level, we would end up with significantly higher, like 10 times higher sulfur levels than we have now, probably. So maybe not 10 times higher, but we'd certainly be 150 or so total sulfur, whereas we're quite low. And that's just us being, waiting for that first edition for a very long time. Like I said, it's mid-May, and many of our barrels have yet to receive any sulfur. So the longer you can wait before that first sulfur addition, the better that wine deals with not having sulfur. Basically, the more the wine is kind of like learning to protect itself, if that's a thing, and then the less sulfur you will ultimately use. So because of that, though, in particular in our cellar, it's very important for us to be very aware of what is going on in those barrels, in particular like off odors, both like volatility. If you're getting like acetobacter, acetic acid formation, volatile aromas, even ethyl acetate, that nail polish remover smell, and of course, like Micah brought up, Brettanomyces, Brett, which kind of has like stable, like barnyard, sweaty, leathery type aromas. All of these things can happen in during the aging process, particularly actually in barrels. Oak barrels happen to be a good way of aging wine, but they're also very good homes for some of these, especially Brettanomyces. Really likes living in oak barrels. The pores, it just really seems to like living in that wood. So we have to be very aware of what's going on in our barrels, because prevention, right? An ounce of prevention is worth a pound of cure. So we really want to prevent anything from going wrong during the aging process.
Yeah, I kind of mentioned barrel maintenance, and again, to be clear, that was on empty barrels, is that, we're sourcing from places we know keep good care of their barrels, and then we're checking them as we load them ourselves. Actually, Nate and I drive up to the Willamette Valley to get them. We bring them back, we steam them again, and then sulfur them to store. Another thing I'll mention, I've seen this a few times, actually, I think I've mentioned it to you, Nate, is that, it's really important to clean the inside of the barrel, but it's also important to keep the outside clean. So, especially being low intervention here, it's like, if you empty a barrel after you've racked a bunch of wines and you make a blend, and you clean and steam the inside, and then you sulfur it, but like you never took that barrel off the rack and there's a bunch of lees all over that, and you steam and you sulfur the barrel that's empty, to be clear again, and then you forklift it into your cellar to be stored, you have a bunch of these lees that sprayed all over the outside of barrels and or the racks, and those are just getting put back into your cellar. And you hit an alarm on your phone that you're going to check in 6 weeks and you're going to re-steam and SO2 those barrels because you want to maintain them. Well, that's great, but the outside of them isn't clean, which is then going back into your cellar, and that can and probably will grow into something. So you want to be really careful, even if you're not low intervention or natural, that you're really paying attention to those little details, because sometimes people aren't sure how they ended up with VA issues on a certain wine, or how they ended up with Brett in their cellar. But it's just these little things, you really have to clean, it sounds stupid, but from the top down, and make sure everything is clean, everything you're using is clean to make the wine, and everything in your cellar is in a good state, because you just want to set yourself up to make the best wine possible.
On a related note, we've talked about this before because I think this kind of varies geographically, but one of the things that can happen in wine as it's aging, especially in barrel, is the formation of film yeasts. So there's Candida or Pichia, different organisms that can cause a yeast that grows on the surface. I guess a lot of people may be familiar with in sherry, right? There's the flor, Flor, flower, that blooms on the top of the wine. That's a film yeast that's done on purpose to create that aldehydic sherry flavor. But that's not what you want to have happen with, for example, your aging red wines like we're talking about right now. And so checking for those film yeasts, which often again are a result of not having fully topped up barrels. If you have only partially full barrels, that's when you're going to get film yeast to grow. And there are various techniques for getting rid of that.
Are you setting me up for something?
I am, Micah.
Awesome, thank you. So we were just talking about how, I think we both worked in places where we've seen this, where someone might look inside a barrel right before they're going to top, or again, if they're taking out X amount of wine because there's a tasting. And I've been in cellars where people are like, oh my God, what is that growing on the top of the barrel? And they immediately, their instinct is let's overtop this barrel and the lot in a lot of cases, right? And let's overflow these barrels that are all stacked in a cellar so that we get whatever is growing on top up and out of the barrel so that it's not growing on it anymore. And then this barrel is overflowed onto other barrels with whatever was growing in there on top of the wine. And then they seal the barrel with a bung that's touching the top of the wine that's inside of it. And even if they hose everything down, even if they use KMBS and tartaric acid or something, you've still just gotten everything that was growing in that barrel all over the place. And I've seen this happen multiple times. I can't stand it. And what we found in our past that has worked is if you see something growing on the top, much like fermentations when we were talking about people smelling or seeing something that looks off on top of something else that's fine, it's don't mix it, don't overflow it. If you're going to do an SO2 add soon, do it sooner. Do it that day, right to the top. Don't mix it in or anything. Seal the barrel, and I bet you're going to open it back up in a week when you're going to top, and whatever was growing on top is going to be gone, and you're not going to smell anything again. So there's not a reason to create all these situations where you're spreading a bunch of things that you see that are growing and could be bad. Just a PSA to kind of think a little more creatively in your cellar and stay clean.
All right, so to kind of wrap up the aging portion here, because we are, again, the purpose of this podcast is to kind of talk about different additives that different people may use during these processes, and to just kind of better educate people why some of the things that go into more conventional winemaking and how Troons may be a little bit different than that. But during aging is another time that people might add tannins. Tannins do come from the oak barrels, for example, a different type of tannins. But if people are aging in stainless steel tank, for example, or if they just are using older barrels and they're trying to get more tannin into their wine, you can also add tannin. You can buy tannin in a bag and add that. There are different products you can add to help control the growth of bacteria or Brett, actually Brettanomyces, a yeast in particular, outside of just sulfur. Actually, Brettanomyces doesn't really care that much about sulfur levels. It's a yeast, not a bacteria, and so sulfur is, not super successful at controlling the growth of Brett and having those kind of off characteristics from Brett. You can add products. Chitosan is the most common one. It's a product that comes from it's actually comes from a different bacteria, Aspergillus niger, and that can help control some of the growth of Brettanomyces. There's lysozyme, a different type of enzyme that basically controls the growth of certain types of bacteria, gram-positive bacteria, which is a lot of the lactic acid bacteria out there. Lactic acid bacteria, we like one of them, Enococcus. That's one that produces malolactic fermentation, but there's other lactic acid bacteria that can be problematic. So there's different ways that you can add things to try to help prevent problems during aging. But at Troon, our method is just paying attention. So if not to oversimplify, but if you can see problems before they happen, you don't have to fix them. And so that's generally what we try to do.
I'll also add that not everyone has that luxury. We've mentioned we
scale.
did, what was it, almost 70 tons this last year of fruit. So, depending how many vessels you have from past vintages in your cellar, sometimes it's a little bit harder for the actual winemaker and assistant winemaker to walk around and taste things regularly. But if you can kind of figure out your perfect scale where you can do that, it will help you not use quite as many adjustments and products.
Yeah, so once the aging is done and you're kind of getting ready to to start thinking about bottling, the next step then would be the blending and the stabilizing of those wines, kind of pre-bottling. So blending is where you're going to, so even, I guess I should make the clarification. So all wines are inherently a blend of some kind, because it's, like if you've got 10 barrels, like we said, all of those barrels are going to be slightly different. And even if they're all the same thing, you're going to be blending them together. So all wines are blends, but sometimes a lot of Troon wines are blends of different grape varieties as well, right? GSM, Grenache, Syrah, Mourvèdre, like red wine type blends. Those are obviously blends, but
some of our rosés.
Yes, our rosés, but kind of all wines though need to be, quote, blended, is the point I'm getting at, is because you're going to be combining wine most likely from different vessels. Like chances are you don't have one giant tank that that all of the wine has been sitting in this whole time. So you're going to have to make decisions about which things you want to go together.
Yeah, and even if the majority was in one vessel, there are little tweaks towards the end you can make. Typically at a lot of places, you kind of have some idea of where different lots or, vineyards in some cases are going. But then you go through different stages of blending, which we actually have another podcast about. So if you're more interested, you can also listen to our exact blending strategy here at Troon. But you essentially will start, maybe a few months before you're going to bottle X amount of wines. Let's say you're bottling, 5 wines in July. And so you will start a blending session with things you kind of know that are going to go together, and some things that maybe you're not sure are going to be like that final barrel that goes in blend 1 or blend 2. And it's usually a good idea not just to have one blending session. I mean, I've been at places where it's kind of last minute and for whatever reason, you need to make kind of a quick decision. But here we usually have, I would say at least like 3, right? I think we've had even more. But you'll kind of do these rounds of, again, it's a combination of like, okay, we have these results and we kind of know how many cases of such and such wine we need and how are these going to fit together, and then what else could possibly go in this blend, or does it make blend A better than blend B? So this is really part of the fun part, is deciding exactly where all of these vessels fit in perfectly with what you're going to bottle.
Yeah, and so we've got another podcast about blending, so we'll kind of skip past that. But once you have the blends put together, that's when you finally know like what your final chemistry is going to be. On that, you finally know your acidity, your alcohol, things like that. And this is where a conventional, more conventional approach would be to do any last minute adjustments to the chemistry. In particular, there may be like a last minute acid add to nudge your acidity a little bit higher. Most places that do add acid, know that like the more they can add on the front end, the better. Like if you can add it to juice, the addition sticks out much less. When you're tasting wines that have had late acid additions done to them, you can kind of taste that acid, like it kind of sticks out to me. So hopefully that's doesn't need to be done. We don't, of course, do them at Troon. We don't add any acid ever. We don't add anything to our wines, as I've mentioned. But this is when you would do that in a more conventional setting. It's also when you would start doing, now that you have your final blend, is when you'd start doing trials for fining agents. Fining is a pretty common practice in red wines. Generally, this is using a lot of animal-based proteins. Actually, fining is the step that makes a lot of wines like not vegan. So when people talk about like, is this a vegan wine or not? A lot of the fining products are animal-based. So common ones that have been used for centuries are like egg whites, casein, which is derived from milk, basically. It's skim milk, basically. Gelatin, from hooves and whatnot. Isinglass is like fish bladders. So like kind of some weird stuff, right? Like why anyone thought like, you know what, I'm going to grind up some fish bladders and put it in this wine and I think it's going to taste great. Like, I don't know, like who was that first person who did that? There's a lot of things you can try. And the reason that people do fining in red wines is generally tannin modification. You can try to, tannins can be both astringent and bitter, and what fining agents do is proteins will bind to tannin basically, and settle those out. So different types of fining agents will bind to slightly different tannins, and then you can set up trials like bench scale in a lab where you add different amounts of these different things. And you can see basically what it tastes like and if you like it better or not. I should also mention that there's actually, going back to the vegan thing, there's a lot of really cool now plant-based proteins that people are using, pea proteins and other ones that come from just other plant products like potatoes and stuff.
Non-fish options.
Non-fish options. Not just because they actually maybe care about their wines being vegan or not, but they've actually found that they work better. They're less impactful. They don't stick out as much. But
And Nate mentioned acid adjustments and kind of like last minute adjustments of the final wine, because once you actually rack everything into a vessel to be that blend that you came up with during your blending session, you always taste it because it might, first of all, taste slightly different than it did in the blending sessions once it's all together. But it's also when you're going to make one of your last SO2 adjustments. So, you'll probably check this again depending on your cross flow and filtration choices that you make, but this is a good way to actually see the makeup of the total final wine for sure. You're not pulling from different barrels trying to find out the chemistry. It's all in one vessel. And you also can make some choices during this period. I won't go too much into detail on a bunch of different products, but earlier we were bringing up instead of using barrels, you could just later use a product. So, if you choose to go that route, you could even a few weeks before bottling just add in liquid form something that substitutes for barrels into this tank. You might have seen Mega Purple before. Aroma gum is often used. So you have a lot of different options if you are in a more conventional winemaking setting where you can kind of play with mouthfeel, flavors, and once you have that final blend is when you want to do that, because you don't want to be taking like bits and pieces of samples from different vessels. It's better to have it all in one.
Yeah. So, and then once you're also in this, vessel, you've got, things settling, things clarifying, so you've kind of gotten to the end of this process. You've blended the wines. You've quote, stabilized it, maybe, done some of these fining trials and, added some other, additives like the Mega Purple or the aroma gum or sugar sometimes, especially with kind of cheaper red wines. If you have something that's really bitter and you don't want it to taste bitter, you can add sugar, and then it won't taste bitter. So, that's something that might be done at that point. Of course, if you add sugar, that inherently makes the wine unstable, because that could start fermenting again. So that would lead you directly into the next step for many people, which is filtration. And filtration can be used for a couple different reasons. The first is just aesthetic. Like, if you filter a wine, you remove any cloudiness to it, if there was any to begin with, depending on how strong of a filtration you use. But then there's also like stability issues here, in particular like microbial stability. If you use like very tight filtration, you can filter out certainly yeasts, which are significantly larger than bacteria, and that would be the thing that might start fermenting in bottles. So especially if you had any remaining sugar, either because it didn't fully finish fermentation, that's more of an issue with or consciously, right, with certain white wines that are off dry, that have residual sugar. Or in red wines, some of those kind of cheaper, more bitter red wines that you've added sugar to make them taste better. You don't want them to re-ferment. You might need very tight filtration to remove the yeast and even the bacteria, to remove the opportunity for spoilage when it's in bottles. So there's a couple different reasons that you might want to filter something. Some common methods of doing that would be plate and frame filter, which are a series generally of pads of different like thicknesses, oftentimes different sizes, with kind of a different. You might do rounds of filtration, kind of getting successively tighter filtration. If you tried to go from 0 to 60, if you tried to just go for like really tight filtration right away with pads, there's a high probability that you would just clog the pads, like you were trying to force too much material through too small of an opening, and nothing happens. The pads just clog up. Pad filtration is fairly simple, but it's also very hard to do well. It's pretty easy for even like home winemakers and things to do themselves. It's like I said, it's pretty simple, but it's hard to do, and it's often kind of impactful. It often leaves like a flavor of a lot of these filters are made out of like paper type products, fibrous products, and it can kind of leave a paper taste to the wine sometimes, especially if the pads were not sufficiently prepped before the filtration began. You can soak them in acid, like give them an acid wash and kind of rinse those through. It can be hard to do. So, that's one way. There's cross-flow filtration, which is very commonly used today. That's a more like technological approach that's a little more modern, at least in terms of its use in the wine industry. It's been used for in other industries since like the 70s and early 80s. I guess it was really the late 80s, I think, that it really started getting used at all in wines. Cross-flow, as the name implies, the wine is flowing across the membrane that is being filtered, as opposed to like in a pad filtration, that's a depth filtration where the wine is directly basically being pushed through the pad. Cross-flow is more gentle in that the wine flows past the cross-flow membrane, and then wine kind of crosses that membrane without being kind of forced through it.
Yeah, and so for these filtration processes, one thing to note, a nominal filter will remove most particles, so that would be like a cross-flow filtration system. And then you have absolute, which is the pad or also sterile filtration. And so you can kind of use a combination if you're choosing to filter your wine. But with both of these, we keep stressing that there's like this final blend and this final tank. And so that system, whatever filtration system you're using, is taking out that final blend. Once you've done everything, you've made all adjustments. The other thing is, like, if you're using products, you want to check and make sure that you leave enough time and you're using the right rate so that you're not clogging this filtration system. A lot of the time, especially for cross-flow, it's like you do it the day before or a few days before you bottle. So it is kind of a stressful time bottling. It's like nothing can go wrong. Like, you have all your supplies there, you're ready. And so you want to make sure that you can actually fully filter your wine from that one tank into another one. You do one more final reading for sulfur typically, just to make sure that it's at the correct free sulfur that it should be at, that you want it at before bottling. And then essentially at that point, it's ready to go for bottling.
Yeah, and so the last type of filtration, which would probably be like sterile filtration.
Yeah.
This often occurs like at bottling, like in line, as the wine is being sent to bottle, because if you are needing or feeling the need to sterile filter your product, and of course sterile filtration, as Micah mentioned, that's an absolute, so it's like a 0.4 micron, very small particle size. And that's an absolute filter, meaning that nothing can get through that 0.4 micron filter. So you know that your wine has no yeast or bacteria in it at all as it reaches the bottle. That's why you do want to have it happen in line. Is that so? Because if you filtered it into a tank and then it sat around for a couple days before it went into a bottle, well, it's no longer sterile, right? So
yeah, and thank you for pointing that out, actually. So a lot of the time, Nate mentioned one way of filtering, and then I followed it with cross-flowing, but it's like you'll kind of pair this, and again, it depends on what your program is, but you will have your cross-flow filtration, for example, a day or two ahead of time, and then you'll sterile filter day of into the bottling process, basically, like on the bottling line. And you could also possibly use a few other things. I guess I'll mention, like, some people adjust, they remove dissolved oxygen. So there could be a few little other like minor tweaks that happen within this period. But essentially you want to choose whatever filtration you would like and do that within the correct window paired with your bottling.
yeah, and bottling then would also, obviously, as it's going to bottle, it's the last time you have to add anything if you are going down the additives route. Again, to be clear, Troon does not fine or filter our wines here, but for others who do make those choices, wines, especially wines that might have some residual sugar in it, the last additive that we wanted to discuss is Velcorin. Velcorin is yes, there's scowls in the
there
room.
Are faces
just
being made.
bring up that word. So Velcorin's the trade name. DMDC. It's dimethyl dicarbonate is the material, and it's actually quite commonly used in many other aspects. So like a lot of sports drinks, that are just sitting there on the shelf. Not being refrigerated with quite a lot of sugar in it and are not exploding. That's because they've had something done to it to make to make fermentation impossible. Because yeasts are everywhere, right? That's what we've been discussing. Like if you leave something with sugar around, it'll start fermenting eventually. Like yeasts want to ferment things that they find out in the environment. So Velcorin is a way of preventing this, and so DMDC, it's very highly controlled because it's an incredibly nasty compound to human beings before it's used. The the reason everyone's like grimacing here is that, so to even use Velcorin, you have to have a tightly controlled dosing system. Is really important to get the dose of Velcorin correct into the bottle. And what DMDC does is it like enters into every cell that's there, so like all the yeast or bacteria that might be there, and it just inactivates enzymes like required for cell function. So it basically just murders every cell, like as it does that. And so the concept here though is that DMDC actually breaks down to methanol and carbon dioxide. So the idea is that this is not considered an additive because yes, it's being added, but all of it goes away. At like they'll even tell you at 50 degrees, it takes 4 hours for the DMDC to completely transform into methanol and CO2, and at 68 degrees, it only takes 2 hours. But I think generally people are like, rule of thumb, if you're using Velcorin, like under no circumstances should you drink this for at least a week, kind of thing. And it's like, and this is so crazy that you would put a product that toxic, right, like into what should be a kind of natural product, one would think, like wine. But the FDA says that this is, the quote is, it's a direct secondary food additive. So it's not required to be disclosed on any label.
I sadly worked with this once. And basically, so what we had done is we had already cross-flowed, and then in line the day of bottling, the wines would go both through this machine, which would dose with Velcorin, and then it would go into the bottling line, which also had the sterile filtration. And so all three of these things are just, preventing against any type of microbial spoilage. And, if, again, depending on your program and how much attention you can pay to your wines, like if anything is left behind that shouldn't be, some combination of these can be used to prevent against any spoilage, right? But again, if you can choose methods that allow you not to use something like Velcorin, for example, that would be ideal.
Well, like abundance of caution, I think, is like why people would choose, especially to do like all three of these, right? Like crossflow and Velcorin and sterile filtration. We were just giving a tour the other day, and we were talking with someone, and what's interesting about it is, at the end of the day, I think sellers that know that they're going to do all of these things, it actually just inherently, and I don't mean to like pick on anybody in general, but it makes you feel a little more lax about certain other aspects of like cellar, like cleanliness and hygiene and things like that, because you kind of know at the end of the day you are just going to nuke that thing. Like nothing is going to survive this end process. So it's like, you've got much greater guardrails on the rest of your process, because you know that no matter what happens in the cellar, you're doing this thing to it, and like that's it. Like nothing more is going to happen.
Nothing's going to survive that combo.
Right, but
including flavor.
Oftentimes, yes. Certainly, like terroir, like certainly soul, like typicity, like sense of place, like that. It's really hard to survive that. But if you're not doing any of those things, like Troon, like you really have to pay so much more attention through that whole process, because there's no safety net later, right? Like, using those products, using, those kind of techniques, that's a huge safety net. And a lot of people, especially with like really massive distribution, if you're like shipping your wine like halfway across the planet, like, you could understand why people like that would have an abundance of caution over the stability of those wines, because like they're going to be on a container ship for months, possibly. Like, they really need a certain level of stability to those that, perhaps their artisanal, distribution model just doesn't need. But knowing that you don't have that as a backup, that really forces you to just be like so attuned to what's going on at every step of the production, every step of the wine through that journey, like right away, just even going back to how we started, that day of picking, really, well, before that, quite a bit. We're at a state vineyard, we get to see our grapes every day. But from the picking decisions all the way to bottling, just every step of the way, knowing that you need to do everything in your power to try to prevent issues from happening, so that you can be as minimal in terms of the use of these additives if possible. Like all of these additives came up to try to help winemakers have more control over their product and more control over the stability of their product, and particularly if things go wrong, ways of fixing it. But humans have made wine for like thousands of years without any of these products, and so kind of having that mindset at the outset and knowing that that's the intention for your wine, and then taking all of the steps necessary to try to avoid utilizing everything we've just spent the last 2 hours talking about. That's of course what we do here at Troon, and I think that really could be a goal for many people. It's not, obviously we do it, it's not unattainable.
I'll also mention, though, you said it a second ago too, it's like it is very possible for people to create a program where they are tasting, they are maintaining their barrels, or however they're storing their wine, no matter what vessels they are, they're keeping up on their SO2 ads. And even if you are using caution and have a ton of experience and your goal is not to add anything and not to filter, again, as Nate mentioned, that's what a lot of these were created for. So there are times that you kind of have to navigate vintage to vintage and things happen. We know that. We've been making wine for a while. But I think just as a little bit of an inspiration, it's possible not to add so many things and not to have a goal of adding or filtering in such a way.
And that is the perfect segue into our now that we've talked about our previous podcast on white winemaking, this current podcast on red winemaking, and I guess I mentioned it in the other podcast, but this actually started a little bit as a joke. Like, we were going to be recording this on April Fool's Day, and we thought, hey, wouldn't it be funny to do a whole podcast about all these additives and just say, hey, just kidding, we don't use any of them. But then we realized that I don't know, there's not a lot of people in the industry who really talk about all of these manipulations that are very commonly done in wines, oftentimes because they don't really want consumers to know. Like, they think consumers won't understand, or they'll be scared off, which both of those things are probably true. So we thought we would talk about it, but from the perspective of obviously we have our own preferences for how we make wine here at Troon. But as Micah was mentioning, there are times when some of these might be necessary for certain people, and we just kind of wanted to go over some of those reasons. But our next podcast will actually be more specifically tailored to the winemaking at Troon, and then how we do, or basically, as you've just learned, how we don't do all of the things that many more conventional wineries employ on a daily basis. So, hopefully you found, I'm sure if you made it this far, you must be pretty deep in the weeds, so you probably found this pretty interesting. And our next podcast will just give a little more clarity onto the process that we use here at Troon.
We are happy to share this podcast from Troon Vineyard, a Demeter biodynamic and regenerative organic certified winery in Oregon's Applegate Valley. We farm like the world depends on it by growing produce in our gardens and naturally crafting authentic wines. We will be sharing these in-depth podcasts several times a month. To learn more, I encourage you to visit our website at troonvineyard.com and those of the Regenerative Organic Alliance at regenorganic.org and Demeter Biodynamics at demeter-usa.org. Thanks for sharing our voyage to regenerative agriculture with us.